Ecologists often need ways to describe and measure how diverse ecosystems are, both within small areas and across larger regions. To do this, we use several related measures of biodiversity at different spatial scales: alpha, beta, gamma, and zeta or zeta diversity. Each captures a different aspect of how species are distributed over space. Alpha diversity is the local diversity within a single area or habitat. In simple terms, it tells us how many different species live in that particular place. For example, if we study a meadow, we might record all the plant species growing within a small square plot. The number of different species in that plot is used as a measure of the alpha diversity of that meadow. In practice, we can collect data using methods such as quadrats or small square plots for plants, transects or straight survey lines for animals or plants, and traps or nets for insects and small animals. After identifying the species found, the simplest measure of alpha diversity is species richness, or the number of species present in the sample. For example, plant species richness is often presented as a number of species per meter squared, with the highest values ever recorded being 89 species per square meter, and calcareous or chalk grasslands, such as that shown here, typically with anything from 30 to 50 species per square meter. Sometimes we also consider how abundant individuals of each species are relative to one another, and so how prevalent they are at the site, calculating values of richness that are weighted by the relative abundance of species. Once we know the diversity within individual sites, the next question is how different are neighbouring sites and communities from one another. This is where beta diversity comes in. Beta diversity measures how much the species composition changes between locations. In other words, it captures the degree of species change from one place to another. For example, imagine two nearby meadow plots. If both plots contain almost the same species, the beta diversity between them is low. But if each plot contains many different species that the other lacks, beta diversity is high. But if we compare the meadow with a nearby woodland, we would expect beta diversity to be high, or in other words, for there to be many species that are only found in one or the other habitat. Curiously, some species can be found in different, highly contrasting habitats. Let's take a very specific example of chalk grassland and neighbouring oak hornbeam woodland. The meadow includes a range of species that are found there but not in the woodlands, such as the green-winged orchid, Anacamptis morio, or the early spider orchid, Ophrys sphygordis. And the woodland includes species that are not found in the meadows, such as the tree species, hornbeam, Carpinus betulus, and Quercus pubescens, downy oak. But also herbaceous species such as periwinkle, Vinca minor, liverleaf, Hepatica nobilis, or Carex alba. These differences increase the value of beta diversity. In contrast, some species do survive in both habitats, such as cowslip, Primula veris. Hairy violet, Viola hirta, and blue bugle, Ajuga or Ayuga reptans, depends on your accent. These species lower the value of beta diversity. So beta diversity represents the balance between shared and unique species. High beta diversity often indicates highly contrasting habitats or strong environmental differences. For the record, the strong environmental difference operating in this example is the annual mowing and maintenance of the meadow by humans, in contrast to a lack of mowing, or at least a different kind of management, which allows the growth of large woody plants and so the development of woodland. Gamma diversity describes the total biodiversity within a larger region that includes many habitats or communities. For instance, the valley and mountains where this meadow is found also includes various types of woodland dominated by different tree species. Beech woodland, for example, dominated by Fagus sylvatica, and also other herbaceous habitats such as fens near the lake and river. Each site has its own local species list or flora, but when all the species from all sites are combined, the total number of species found represents the gamma diversity of this geographic area. So gamma diversity encompasses multiple sites or habitats, but must always be defined relative to a specified geographic region. That region can vary widely in size, such as the gamma diversity of one particular mountain, of the entire mountain range or even of an entire country or biome, depending on the question and scale that we're investigating. So gamma diversity reflects the overall species richness of the landscape, ecosystem, or biome. It gives us information about how biologically rich an entire region is. To measure gamma diversity, we gather species data from multiple sites across the region and combine all the records and count the number of unique species present. However, when we're comparing habitats, beta diversity compares only pairs of habitats, one against the other. But the overall landscape and gamma diversity are composed of more than just two habitats or pairs of habitats, and we need a way of comparing multiple habitats together. So zeta diversity is a newer concept that looks at how species are shared among many sites simultaneously. The idea is to examine how many species occur across increasing numbers of locations. We describe this using different levels or orders of zeta diversity. First order zeta diversity represents the average number of species in individual sites, similar to alpha diversity. Second order zeta diversity measures the average number of species shared between pairs of sites. Third order zeta diversity looks at species shared among groups of 3 sites, and so on. As more sites are considered together, the number of species shared by all of them usually becomes smaller. This pattern helps us to understand how common or rare species are across a landscape. Species that appear in many sites contribute strongly to higher order zeta diversity, while rare species appear only in lower orders. To measure zeta diversity, researchers use a species by site table, which records whether each species is present or absent in each location. Computer programs then calculate how many species are shared across different combinations of sites. Because the number of possible combinations can be very large, we have to use specialized statistical software to estimate these values efficiently. These 4 measures provide complementary ways of understanding biodiversity. Alpha diversity describes the variety of species in a single place. Beta diversity shows how much communities differ between locations. Gamma diversity describes the total diversity across an entire region, and zeta diversity examines how species are shared across multiple sites at once. Together, these tools allow us to build a clearer picture of how life is distributed across ecosystems. This information is essential for conservation because it helps identify areas with high biodiversity, understand how habitats differ and how they can be similar, and to detect changes in biodiversity within and across ecosystems over time.